Thanks so much for the kind introduction. It was really a nice surprise, and I'm really happy that you were the one introducing. The only thing that I have to say that is, unfortunately, my position in Würzburg is not tenured, which means that I'm currently job hunting, and for that it's even better to have such an amazing audience like today. So thanks for this opportunity. And in the next 20 minutes, I want to take you on a short journey into the nano world, and in particular into the world of drug-loaded nanoparticles. And I would like to show you how, on the one hand, we use NMR spectroscopy, but also combine it with different complementary tools, for example, from X-ray diffraction, from calculations, also together with some pharmaceutical groups. And I invite you to take a look behind the scenes and see what we can find out with NMR spectroscopy. So first of all, before going to the science, I have to say thank you, because I have a really wonderful team here. I have the first PhD student graduating at the end of last year. He handed in and now he defended. And I have this wonderful team who are always like really motivated, hardworking on the topics, and who are also bringing happiness and not just working hard, but also having a good time. And another thing that is really crucial is that I have so many really cool collaborators who have very, very interesting research questions and always come up with new and exciting things to have a look at. And as Adam already mentioned, I mean, without support, this would not happen. Or a context. So if you think of the challenges that the pharmaceutical industry is facing, and this is not just in the last years, but for decades, it's one of them is low aqueous solubility of the compounds that are developed. And to give you a few numbers, around 40% of the currently marketed and 60% of the compounds under development show poor aqueous solubility. And this can lead, for example, to reduced bioavailability, or, for example, also to variability between patients, in between patients. And there are multiple ways how one can deal with this issue. And I would only like to focus on one particular one. There are physical methods, physical-chemical ones, and I would like to focus on polymer micelles, and in particular block copolymer micelles. So you have a polymer which consists of 3 different blocks. The outer ones, here shown in blue, are more hydrophilic, and then the inner one, shown in red, has more hydrophobic repeating units. And then if you have these strands of polymers and they are in solution surpassing a certain concentration, the so-called critical micelle concentration, then they can self-assemble into particles in the nanometer range. One example is shown here. And what we typically assume is that the hydrophobic parts form the core of this particle. For example, also allowing the transport of hydrophobic molecules or cargo in the center. And then the hydrophilic parts that you can see here as this fluffy hair, they are providing solubility and also protecting the cargo. And this is the ideal image that we have of such particles. And this ideal image is not just something that I have made up, but it is actually prevailing throughout the literature. So you can see here multiple examples from the last 10 years in the literature. And in all of them, you see this same distribution of tasks, with the loading taking place in the center and then the hydrophilic corona on the outside. And only in recent years, I would like to highlight in particular the work by Martina Stenzel. There have been more detailed investigations on the molecular level, also correlating, for example, the loading with specific features, such as how is the uptake into cells. And if we go to this molecular level information or get it, then it means that we can more specifically tailor how we want to change a specific system for an in-mind application that we have. And this is where we would like to contribute with our work group. Of course, to contribute in this area, it requires not just a small puzzle piece, but actually many, many pieces that have to be coming together to improve the solutions that we have in drug delivery or in medicine in general. But we think that improved structural understanding can be one such small puzzle piece and can work really well together with other information that we can gain. And today I would like to show you some of the work where we try to get a better idea on how these particles look on the molecular level. And in the second part also to show you what happens if we now dissolve these molecules in environments that are more closely resembling what is happening in our body and how they behave in intestinal fluids. So first of all, I would like to introduce you to the polymer platform that is developed in the Luxenhofer group. So Professor Luxenhofer, shown here, is professor in Helsinki. Previously, he was at Würzburg University, and they are making these polyoxazolin triblock copolymers. And they are quite remarkable because in contrast to the currently available polymers, they can encapsulate almost twice as much as, let's say, the standard that is available on guest molecules. And we chose one guest molecule that they have worked with before, curcumin, the natural product, and it has the advantage that it allows very, very high loadings on one hand, and it also doesn't overlap in any of the carbon spectra in the NMR data. So it has multiple advantages, and so they provided us with a set of formulations of different amounts of curcumin within the polymer, ranging from 17 to more than 58% of the guest molecule. And I will term them low, medium, and high loading from now on. So this leaves us with the first question: why is it possible that these polymers can accommodate so much of the guest molecule? And another thing is that we can look at properties or pharmaceutically relevant properties, and one that we did look at was the dissolution properties. So we pressed tablets of these formulations and then dissolved them in a serious dissolution apparatus. And you can see here for the low loading in gray that the dissolution takes place almost immediately. And after 3-4 minutes, everything is done. So the whole sample is dissolved. If we go now to the medium loading, we have a lag time, then a dissolution, and then we have a quite variety of end states, let's say. And then it gets worse if we go to the highest loading. So we really have to compromise the dissolution of these particles of the sample. For the highest loading, you can see that even after much more than 1 hour, there's only a part of it dissolved yet. And this led us to the second question. So why do we have this compromise in the properties? And the first idea would obviously be to try and measure NMR in solution, because strictly micelles are only existing in solution from the definition. The problem is what you see here. If we dissolve everything in chloroform, where all the parts are individually soluble, you can see that there are peaks for the polymer, which is green and orange, and there are also peaks in blue and gray for our guest molecule. But as soon as we have micelles in aqueous solution, there's nothing in this area. So the core of these particles behaves almost solid-like, and if we would increase the temperature for quite a bit, we would see very broad features in the baseline, but we cannot really do much spectroscopy such as NOE with these signals. So this is why we went to solid-state NMR, and we freeze-dried the samples from the previous slide and subjected them to solid-state NMR measurements at moderate magic angle spinning. And if you look at the bottom, you can see in blue the pure polymer, in gray the crystalline guest, a 1-to-1 mixture just obtained by grinding. And you can see that it's a 1-to-1 addition of the gray and the blue spectrum. However, if we go to these yellow reddish colored samples, they are actually the 3 loadings from the previous slides. And you can see we have broader signals, we have changes in chemical shift, we have increasing signal intensity for increasing loading. So there's lots of stuff that we can read from these spectra. And you can also see from the exemplary one in the top that the spectra are more similar to amorphous curcumin than they are to the crystalline compounds. And this is also confirmed by X-ray diffraction. So we have throughout the whole measurement also nothing induced by MIS. We have an amorphous sample, and it can be stored in the solid form for multiple years without crystallizing. So let's take a look what we can actually learn from the measurements. And we didn't just do the carbon data that was shown on the previous slide, but also proton data at slightly faster MIS. We measured proton-carbon correlations, and we combined it with CASTEP calculations. And what we can see are 3 things regarding the guest first. So if we increase the amount of guest that is in these formulations, we can see that it gets less ordered. And this would not be necessarily expected, because if you think that you put more and more guest molecules in a specific space, it could also lead to crystallization nuclei and then finally to crystallization of the compound. And this has not been observed since we started working with these molecules. And combining the NMR with the calculations, we could see that these polar groups at the outer sphere of the molecule are actually the main responsible coordination partners. So they are the hydrogen bond formation partners. And what we can also see from the calculation is that the molecules inside of the polymer matrix are largely planar, while the crystalline compound, or the most stable form, is actually twisted along the axis of the molecule, as you can see here in this green extract. And we were also able to synthesize derivatives of this curcumin molecule, which is not yet published, is still under construction. And we could show that if we make these OH groups unavailable, then our maximum achievable loading drops significantly to less than 0.1 from more than 10 grams. And we could also show that if we take this enol functionality away, which is not contributing significantly to the coordination to the polymer, then we still have a high loading. And we can also now switch and look at the polymer. We have tertiary amides in the polymers. And this means there's no hydrogen bond donor whatsoever in the polymer, so it can only accept hydrogen bonds. And the most important experiments here were the head core, so the correlation experiments, where, no surprise, we see proximity between curcumin and the amides of the polymer. But what was really surprising is this one here on the right. We saw a close intermolecular proximity between the methyl groups of the outer hydrophilic blocks and the curcumin. And this is also mirrored by changes in the chemical shift and also changes in the full width at half maximum. And this was the first experimental indicator that not just the hydrophobic part, but also the hydrophilic part of our polymer is actually responsible for coordinating to the guest molecules. And this now gives us a possibility that we can explain what is happening here between the different polymer drug assemblies and the properties that we observed. So if we think in the low loading, we have lots of coordination spheres in the hydrophobic core that are available to coordinate the curcumin. But as we put more and more guests inside of these particles, these positions get saturated. And then we need the hydrophilic arms from the interface and also from further away to stabilize if we want to incorporate more and more of our guest molecule. And if you now think in lots of these hydrophilic units coordinating curcumin, it means that the curcumin is fighting with the water molecules for these coordination sites. And if the amides in the hydrophilic chains are not so available for the water molecules anymore, it means that we have to compromise the solubility or the dissolution of these particles. So this was a really cool result because we could derive first hypothesis. We could also experimentally confirm some of them. But what we see here is still a schematic drawing. I mean, we are still not really to molecular level information. And this is when we started to look more closely into evavirans, which is a drug against HIV, also having a very poor aqueous solubility. And I always put this big playground because if I look as an NMR spectroscopist at evavirans, this is what I see. So there are multiple possibilities of nuclei that you can measure in experiments that you can try. So this is like our toolbox molecule with which we try to do interesting new experiments. And in this case, we combined experiments with molecular dynamics simulations, and we combined different through space proximities. So you can do proton-carbon, and you can see that the aromatic side groups are in proximity to the backbone of the polymer. You can do fluorine to carbon through space proximities, and we found that this CF3 group is close to this hydrophobic chain. We can do this with proton-proton and with nitrogen-proton and can then complete to actually show the different proximities. And if you look at the color scheme and where these connectivities are, you can slowly start to imagine how this molecule could lie actually in this coordination pocket. And as you might have seen from the quite amorphous and broad spectra, we have a distribution of environments. And what is better than showing the distribution of environments than molecular dynamics? And we teamed up with Professor Sulzrifer, who is here in the pharmacy department, and he's an expert in modeling proteins in molecular dynamics. So they constructed a micelle out of hundreds of polymer strands and hundreds of evavalence molecules, and then did the molecular dynamic simulations, and then gave us back snapshots, as you can see here on the left, where you can see all sorts of different arrangements or distribution of arrangements of these evavalence molecules within this polymer matrix. And we could also observe similar features in the behavior during the molecular dynamic simulation than we saw in our model from the curcumin project. So we could actually see that for lower loadings, the arms are rotating more freely of the polymers, while they are folding more to the surface if we have higher loading. And what was also really interesting is that they provided us with short distances, or let's say significant or catchy distances. And one that they found as particular from the MD simulations was this short distance below 3 angstroms, which was between the fluorine and the butyl chain that we also identified by NMR spectroscopy. So it seems that the molecular dynamics simulations can be kind of a benchmark with the NMR. And now we are doing more simulations with different polymers, with different amounts of guest molecule to tackle the different features that we observe in our experimental data. So this is ongoing, and with this I would switch a bit to show you also that we're interested in going to more biological environment, because if you think in taking a tablet, then it would at some point arrive in our gastrointestinal tract. And this is much more complex than what we generally test in vitro experiments. And we would like to learn more on this to maybe even be able to shorten a bit the gap between what we observe in vitro and what is then actually happening in vitro. So we are starting with these simulating intestinal fluids, and then we want to go more complex in the future. So if we think where this drug absorption takes place, so we are now in the intestine, and we have a fluid layer, a mucus, an outer and an inner mucus layer, and then we have the step of permeability so that the drug can actually be taken up by the cells. And obviously all these processes, first, our drug has to be released from, for example, our polymer matrix, otherwise it cannot do its action as it should be. And we were introduced by the pharmacy department to these mucus studies. So we are also doing some work with mucus, but today I would like to show you what is happening in our intestinal fluids. And there is one company based in the UK called BioRelevant, and they give different media simulating the fed state or the fasted state, so after or without uptake of food. And there is a certain number of compounds with a specific pH and also salt concentrations. And the main component here is taurocolate, an amphiphil, which is essential in solubilizing lecithin. And there's not just lecithin, but there are other compounds like oleates, or we have maleic acid as buffer. So we have this whole system, and the molecules that you find here on this page, they are actually doing a small aggregation party between themselves. So they self-assemble into micelles and vesicles. And if we now add different components from the outside, we can actually see what is happening, or how these aggregates and colloids that are present there are changing. So you can see here from cryotem data that the resolution is not so good because of the large amount of salt that we have inside of our samples, but we can distinguish a profile that we have around 5 nanometer sized compounds, aggregates. And now we can add polymers or drugs. And I have shown you here 2 different examples. The first one is commercially available. So it's present in many cosmetics and drug products already FDA approved. And the second one is the polymer the oxazolin that you know from the previous data that I've shown to you. And if we throw now different amounts of polymer into these passive mixtures and just observe what happens to the proton chemical shift, we can see that for the commercially available polymer, there's some shifts being observed, while there's almost nothing observed in case of these oxazolin polymers. And this might indicate that on the one hand, we have something interfering with our bile salts, and in the other hand, we have something that is non-interfering, at least if we just add the polymer itself. And then we also added the guest molecule, so the evavirans, the HIV drug from the previous topic. And we measured predominantly diffusion NMR. We also did NOE, we did hetero NOE, and we also did cryotem for all of the samples. And for the diffusion data, the biggest challenges were to find signals that unambiguously belong to the specific components and then appropriately fitting the decay curves that we found in the diffusion data, because they were not always mono-exponentially, but below the 1% decay limit. They also deviated from this profile, and there were some, if we had passive, the polymer and the evavirans, that we could actually no longer fit with mono-exponential decays, but actually had to use log normal distributions. So now you get the two graphs where we have all the diffusion data accumulated for the different samples. And so you see in the colored dots, the diffusion coefficients. And because this is a very crowded graph, I would like you to first focus only on the left side. So we have taurocalate, which is the black. It's soluble in water, so it's showing an average diffusion coefficient between the free and the aggregated species. And then we have the lipids in red and the polymer in light blue. And you can see that they diffuse together. And the more polymer we add, the slower the diffusion, so the bigger the particles that are formed. So this is confirming what we had before, or already guessed before, that the commercially available polymer is interfering with our biocomponents. And now we add the green dots, which is the drug. And you can see that now everything is changing. So we still have the black in the bottom, and we have the red and the green diffusing together. So the lipids and the drug is now diffusing together, and the polymer is doing something different. So the polymer is in larger and more slowly diffusing particles, but not for the whole concentration range, but only until this point here, which is actually the solubility limit of evavirans in this intestinal fluid. And in this moment, if we want to add more of the evavirans, we need the support of the polymer. And from then on, we have a collapse into almost an equal diffusion, so one particle, and then we have again an increase with increasing concentration. So this is already showing that as soon as we add guest molecules, the situation becomes a lot more complex. And for the same for the other polymer, so on the left side, you see again dark blue now the polymer, in red the lipids, in black the taurocalate, and you can see that they are not diffusing together, so they are largely in separate particles as we expected from the proton data. But as soon as we add the drug again, you can see that all of them are diffusing together now, so we have clearly mixed particles as soon as we add a drug. And this is very interesting because it means that we have a concentration and composition dependent behavior, aggregation behavior in these simulated intestinal fluids. And if we would just screen this top part to check for the different polymers, we might miss that there's a really rich aggregation behavior at different concentrations with different compositions. And now imagine how many people you know who are taking more than one drug product per day. And if you have multiple excipients, polymers, and everything in one tablet, it might not just be that the individual drugs are interfering with each other, but it might also be conceivable that the excipient in one drug formulation is doing some weird aggregation with the drug of another one. And with this, I would like to summarize. You have seen this work on getting structure property relations and models for these polymer nanoparticles. And you have also seen the work that we did on biorelevant media. And what we are currently also working on is we study the hygroscopic behavior of these particles and trace water through them. We would like to combine the work that we have seen on molecular dynamics with machine learning, because we have tried to calculate shifts from the snapshots, and it has taken us an immense amount of time. And we have also done first experiment where we tried to encapsulate radicals into the mice cells. And now on this site with the biorelevant media, we have indications that there's also a loading dependent aggregation behavior. I already mentioned that we are with the pharmacy department interested in combining FESIF with the mucus. And then of course, another step would be also to put in the permeability. And there is so much more to learn that we are really excited to have a look at. And so I thank you very much for your kind attention, and I'm really looking forward to the questions. Thank you very much.
Thank you very much, Ann-Christin, for a great talk and also for staying on time. We can now have a lot of questions. Maybe I can briefly start with one myself. So, I mean, in the beginning, you showed this nice solid state NMR spectra. And so if you do protein solid state NMR and you go for proton detection, there's basically two strategies how to improve the resolution. One is to spin faster and faster, and the other one is to deuterate the protons. So I'm wondering if you have a similar plan or strategy to improve your spectra.
Yeah, this is a really cool question, because we have tried spinning faster, and we actually had the pleasure of visiting Bruker before Corona and everything, and we measured not the samples here, but with a more complex drug molecule with the 0.7 millimeter probe. And the resolution above 60 kilohertz didn't improve anymore. So it seems that the intrinsic broadness or distribution of environments is prohibiting that it gets a lot better at higher MIS. But I actually like the idea with the deuteration. So this would be a cool prospect. We've also thought about other isotope labeling, because we have one molecule that is having nitrogens. So yeah, this second approach seems to be more feasible.
Okay, thank you. Next question by Art Palmer.
So this just will illustrate how little I know about biology, but in the second part is the association of the drug with the lipids and the polymer and the mock intestinal fluid. Is that a good thing in that you solubilize more drug, or is it a bad thing because that just leads to excretion rather than absorption by the cells?
This is a really cool point. Yeah, thank you. So the good thing is that this mixture with polymers, for example, in the curcumin case, it actually leads just for the lowest loading to a 6,000-fold increase in the amount of compound that is dissolved. So with this, there is the possibility of getting a lot more to the point where something is happening. But of course, I mean, in the moment where you're adding another substance to this already complex mixture in the intestinal fluids, I mean, there is on the one side the danger that something would just go through and it would not release the cargo. So this is one question. And then there's another question of interfering with an otherwise healthy system, because this is what the bile components are doing. They are taking the drugs and transporting them so that they can be taken up by the cells. So if we add something, we might also interfere with this natural process. And this is why it's so important to have a deeper look. What is interesting, or what we observed, is I showed you in the beginning the system where we don't see any proton signal for the guest molecule. As soon as we put everything together inside of the intestinal fluids, we reobserve this signal for the guest. So this means that in the presence of the bile, it is less tightly bound, or let's say protected inside of the polymer than it was before. So this would be, let's say, a slightly positive indicator, but of course there's a lot more work that needs to be done. So thanks for the point. Yeah.
Okay. Then we have one question in the Q&A. Emmanuel de Rutis, maybe I should read it. Thanks for the talk. I didn't get why solution NMR didn't really work for the first application you introduced. Would you like to comment on this again?
Yeah, so we have done also solution NMR when we have a clear signal from both the host and the guest, so that we can trace different changes, for example. but in particular for the polymers from the Luxenhofer group, we barely see, I mean, if we go to a cryoprobe 600, we can see like a broad baseline at about body temperature, but it's really difficult to get a clear signal for the guest molecules. And then it's also very difficult, for example, to do intermolecular interactions or find out who's close to who if you don't properly see the signal. But for the cases where we see all the, let's say, ingredients in our mixture, we of course go for the solution as well.
Thank you. Then Kai Saalwächter.
Hi, Christine. Yeah, thanks for sharing these nice results. I think my question goes the same direction as Adam's. So you actually freeze dry the micelles, and that means you lose the water not only in the outer shell, which possibly turns glassy, but the question is, may there have been water inside as well, and also lead to a glass transition like phenomenon? In this context, I would think in an amorphous environment, you never expect narrow peaks. So I think deuteration would only be an expensive way to prove that point. Yeah. But actually loss of water even on the interior, which may actually adapt a little water, could change the structure and actually explain the observations that you make. Did you try to rehydrate a bit to see the effect of that?
Yeah, so what we did is that we always, when we worked, so it's a really good point. So we did multiple experiments where we had first, the compounds, maybe no, it's not here. Okay, so if we make these formulations, then they are done by a thin film method. And so they are produced in solution. And we then freeze dry it, and then we redissolve it and observe with DLS and SACs as well. Drying are the same, but of course, that's not a sufficiently good answer. And we also tried, and this is some slides that didn't make it because of the time restraints, but will certainly be shown tomorrow. We started to also take solutions and freeze them in the spectrometer and then measure ice in the solid state and see then how the signals look and compare to the ones that we have from the freeze-dried solid state NMR data. And we also did it the other way around, but this hasn't worked so well because we have too little sample amounts. We have also tried to put the freeze-dried in the rotor and then rehydrate it by only putting 5 micrograms. That's what I mean. Yeah, and this hasn't worked so well, but the ice has been pretty cool, even though the PhD student was pretty scared when he was dealing with the ice. But this has been going well, and we will extend it to more different systems to prove it.
Thanks.
Next question by Francis Sekarovic, please.
So I was wondering if you thought about using DMP to look at functionalized nanoparticles.
Yes. So this is why on the Outlook slide there was already that we played a bit with radicals. So we have recently, but this was only like a short project where we wanted to see how well it works. We tried if we can use differently polar radicals and get them in different compartments of the particles so that we have something that is water soluble and might accumulate more in the shell, and something that is less soluble and might be more, at least at low concentrations, in the center. And it seems that we observe differences also in the relaxation behavior. And then the next step would be EPR, which is scheduled for October, and then let's see. But yes, I would be really curious to try that and would be very happy.
Okay, so Art, did you have another question also?
Yes, on the first part, you had the surprising result that high loadings led to more amorphous character of the drug rather than crystalline. And I missed the connection as to how that explained the slow dissolution of the drug from the particles that you showed in the beginning.
Okay, no, this has nothing to do with the slow dissolution. So sorry for not making this clearer. So the slow dissolution comes from where the molecules are located and who is involved in the coordination, so that the hydrophilic parts are also involved in coordinating our guest molecules, while the amorphous character is something that is a characteristic of all the formulations that we have. So all polymers and all molecules encapsulated are always amorphous. So this is like just if you have, there's a lot of studies on amorphous solid dispersions, which are not forming these type of particles. And in that case, you often have the problem of recrystallization from the matrix. And I just wanted to make the point that for the systems here, we have not observed that.
Okay, so thanks again, Ann-Christin, for the talk, and everyone for the lively discussion.
Yeah, thanks for the questions.
I think we have to move to the second speaker. It is my great pleasure to introduce Julien Wist. Julien studied chemistry at the University of Lausanne, also nearby at the EPFL. He did his PhD under the supervision of Professor Geoffrey Badenhausen. And then later on, he moved to Colombia, where he had different positions, assistant professor, associate researcher, and even director of research at two different universities, in Bogota first, and then in Cali, where he established his lab. More recently, he joined a Murdoch University, I believe, as a visiting professor, where he is now currently working with at the group or in close collaboration with a group of Jenny Nicholson and Elaine Holmes. Julien has an interest, a keen interest in small molecules and in NMR, obviously. He likes developing applications in nuclear magnetic resonance, I believe first in proteins with Jeff Badenhausen. But later on, I'm very often with the small molecules, and that's I believe, for what he is best known. He has a method for predicting chemical shifts that it works. We've used that, and then recently he has shifted gears towards metabolomics. And I must say that he has made very, very significant contributions recently in the COVID pandemic, where he had seminal works by characterizing the metabolism of COVID-19 patients. So, Julien, whenever you want, it's a pleasure to have you here. Cuando quieras.
Yeah, thank you for the nice introduction. And so let me share the screen, and thanks the organization to invite me, and really nice to be with you to share that recent work and recent new passions with more biological data than I'm used to. So yeah, I will just today try to show the recent results we've obtained by revisiting an old observation window or old IDs in search for new biomarkers, and I will try to explain how we actually come to this, why we actually started to revisit those old pulse sequences. But first, just to put a little bit into the context, I've moved recently, so I'm still based in Colombia, but I'm on sabbatical leave at Murdoch University, and the Phenome Center here started like 2 years ago. So it was basically opened a few months before we got hit by the pandemics. So obviously it kind of shaped a little bit the whole research program, and a lot of things shifted into research about COVID-19. So at the very beginning, the first question was about the detection of the infection and the virus, or the direct effect of the virus on the metabolism, and see if we got complements to the diagnostic methods available at that time, very early in the pandemic. Then the interest shifted a little bit and moved into the severity issues, and the idea was to see if it would be possible to predict why some people would actually convert into very severe patients, while others would basically travel through the disease only mildly affected. And more recently, maybe 6 months or a bit more now, the interest shifted again towards trying to understand what happens after COVID, since there is a lot of reports now. I'm seeing new literature literally every week about the fraction or the proportion of people that are still affected and report symptoms 3 months, 6 months, and even 1 year after the infection. So this is to shape or to explain a little bit the concept, but then the hypothesis or our understanding of the hypothesis of how the conversions or the phenotypes conversion from what we call a healthy space into an inflammatory space due to the infection by various works. So we have so our hypothesis is that we have those different people in this healthy space, or what we would call a normal space, since not everybody in the normal population is actually healthy. Some people have comorbidities or cardiovascular risk, diabetes, and so on. So from these, what we call normal spaces, how people actually move, or the metabolic profile is shifted by the inflammations and go through different respiratory or cardiovascular or neuroinflammatory space, or even arthritis we have in that case. And then after this phenoconversion, we are interested in how people actually would come back. And in this journey back to the normal space, some people would maybe shift to different metabolic spaces or phenotypes with a higher risk than they had before, and eventually never really get back, or get back but after 3 months or after 1 year. And in these, what we call this recovery space, we have the symptoms that are commonly reported by the surveys that we are following very closely. And within those spaces, obviously, we are interested in measuring different parameters that will open a window that will shed lights on different aspects on those diseases. So we have on the right side the panel of the markers or parameters that we are able to measure. But today I will mainly focus on what we were doing with NMR, but obviously we are always interested to some more parameters and to add to the toolbox of parameters we are able to measure by NMR, but also by targeted mass spectrometry mostly, and I will explain why. And I think the reason why a lot of work we've done recently shifted from exploratory mass spectrometry to targeted mass spectrometry is because we would like to move very fast into this crisis. So the whole project started very much like all these metabolic phenotyping projects started by recruiting and finding some patients with following actually the infection, so with active COVID infections. We also collected a control cohort, very similar to all the project in that field. And then in order to move quickly, the first parameters that were measured were targeted NMR. So NMR is very quick and robust when we compare that to mass spectrometry. And we had, in collaboration with Bruker, access to the lipoprotein, so IVDR lipoprotein model, where 112 lipoproteins can be extracted from the proton NMR spectrum. And those were also enhanced by a few more parameters measured by mass spectrometry. So the important things here to notice is that once we built model to classify those two populations, very quickly some variable got highlighted. And interestingly, when we especially move to, for example, CPMG spectrum, where we get rid of all the interaction from the proteins envelope, the two signals were highlighted very strongly, GLIK-A and GLIK-B. So I removed the ppm, but 2.03, 2.07 ppm on the right side, and SPC, which is 3.2 ppms approximately. So those signal were very, very strongly highlighted, and one actually to the increase, so mainly increasing when with infection, while the others is pointing downwards, meaning that it's decreased with infection. So this was with a very tiny set of data, as you can see, approximately 20 people in each cohort. So very preliminary results, but already indicating something about the inflammation role in the infection. So then come a second phase of validation, this in collaboration with Oscar Mille, and samples that they sent over. They collected a big cohort that we call here a Spanish cohort. They've analyzed the cohort themselves in Spain and sent the aliquots over to Australia. And we performed what we call twofold validation. So what we refer to analytical validation, but also a biological cross-validation. So it means that mainly we've analyzed the sample on both laboratory and compare the results, and you can see on the middle panel that the scores obtained now with a slightly larger cohort are very reproducible. So basically we can the conclusion is we can merge data, we can cross model data acquired in different laboratory, following obviously the standard methods developed and pushed by Bruker, referred to as IVDR method. So providing we all acquired those spectra using the same parameter sets, we can basically merge the data, and we can see these RMSD plots where we can look at basically considering both cohorts as replicates, find the RMSD for the two groups, so the control groups and the acute phase group, and we can see that the error on the replicates is, the experimental noise is basically much smaller than what we expect to observe. But most importantly, if we look at the loadings again, the loadings are very reproducible. It means that the biomarkers that are highlighted by our measurements at ANPC and the measurements performed at SIG BioGuinea in Spain were very similar and all pointing to these inflammation markers. So this is one analytical cross-validation. And then what we could also do is to use the model we obtained with our Western Australian cohort, use this model and predict the results that should be obtained for the Spanish cohort, and vice versa. We can build a model using the Spanish cohort and then predict the results for the Western Australian, and you can see one of those results where we cross model on the panel. I mean the scores on the bottom part of the slides, where we can actually see that the model is really good, and we can exchange. So one model built with a Western Australian patient is very similar to what was obtained with this Spanish cohort. So this was a first step into validating what we were actually observing. Now, just to explain a little bit what we are observing. So GLIC-A, GLIC-B, and now what we are introducing at SPC are markers of inflammation, or GLIC-A was actually proposed first by Wist, and that's what we refer to as GLIC-A and GLIC-B, with as we can see here, that provides from the N-acetyl methyl group attached to the glycoprotein. And we are proposing to look at this signal that is observed to go downwards, so that decrease with infection, and that we call SPC, which also is a composite signal made of the mobile choline head groups, mainly from the LDL and HDL lipoprotein particles. So this is just to introduce, because I will mainly refer to GLIC-A, GLIC-B, and SPC further, and it's I'm not sure that it's clear to everybody what those markers are. So Oddvost showed that, or made the first link between those signals and the inflammations, and we are trying to, as I will show, make the links for SPC and try to understand how SPC differs from GLIC-A and GLIC-B. So just to summarize a little bit where we are right now, we've just validated and showed that standard experiments, in this case IVDR, allows us to join and to merge a method, a cohort acquired in different laboratories. We also did similar experiments with targeted MS, so we can actually, if we extract features from those experiments, we are able to remove the the errors coming from the experimental conditions. I haven't gone into the detail, but we can analyze samples in 3 millimeter tube, 5 millimeter tube, according to the variability and the volumes of the samples that we have. And then both the GLIC-A and GLIC-B and SPC inflammatory markers are highlighted when we study those COVID cohorts. Now, the question that I would like to develop a little bit further today is how accurately to determine those concentration of GLIC-A, GLIC-B, and SPC, so that we can get something like I show in the lower panel, where we would have the healthy group, the acute phase patients, and we can see that GLIC-A is actually strongly increased, as is GLIC-B, while we can see in the univariate box plot that SPC goes down, and we can use this very useful, and we've shown very significant ratio of SPC over GLIC-B. When we look at the spectra, the problem is a very old problems. We have a very crowded spectrum with a lot of overlap. And if we look at the 1D NMR spectra, we usually refer to as noisy, since it's a 1D noisy experiment. We can see that we recognize basically the strong distortion of the baseline that comes from the contribution of the proteins, mainly albumin. And on top of that, we recognize the GLIC-A and GLIC-B signal, and we can also see SPC. But if we look at GLIC-A, there is a strong overlap with remnant signal from lipoproteins on the right, on the high field. And then if we look at SPC, we also have overlap with small molecules signal from glucose on the left-hand side. If we use another very common experiment in metabolic profiling to complement with the NOSI is using CPMG to remove the protein's contribution and focus on the small molecules. In that case, we see, I mean, panorama is a bit better, but still we have this huge contribution from the lipoproteins coming from the mobile. There is no chance to get rid of this overlap from that point, and then the overlap with the small molecules remains for SPC. So how can we improve on this? The idea was to try to get rid of the small molecules, and there was an experiment proposed years ago by Jeremy when he was working at Imperial College, which they call a DIA for diffusion and relaxation editing spectroscopy, where basically it's an adapted and adjusted pulse field gradient experiment where we can use a diffusion filter to remove the contribution from the small molecules. And then, as we can see, the spectra that we obtain is much cleaner, and we at least remove most of the overlap on the SPC peak, while we still have this nasty contribution from the lipoproteins. So we were looking for improving on those parameters, and to be fair, we had the approach was basically a trial and errors, because we were misled at the beginning, thinking that the driving filter was actually a relaxation, while it turns out that in order to obtain a clean spectrum, we had to adjust our double echo experiment. I will show the sequence later on, to adjust the delay, sorry, in order to defocus completely the contribution coming from the lipoproteins, and trying to select only a singlet contribution in the spectrum. And this latest pulse sequence that we call JEDI, for J-edited diffusion experiment, we combine mainly a diffusion filter with a relaxation filter, and we've tuned the delay in order to try to optimize the selection of singlet and get rid of every contribution from different multiplets. And as we can see, if we look on the gray area on the right panel, this is a spiking experiment where we added to the normal plasma sample with parenteral nutrition. So when patients are received in ICU, some patients required additional so external parenteral nutrition treatment, which was pretty common in the very severe patient we received in our cohort, especially one cohort from the Cambridge University. And we always had the discussion, how should we treat those spectrum, as we were actually measuring patients that were not spiked, but actually treated with a mixture of lipids, amino acids, and sugars mainly. So in that case, we can see how the addition, the spiking with lipids is actually distorting the result from the nausea. And even on the Dyer experiment, we can see a strong contribution from those lipids. While if we go to the JEDI, we can see that also there is still a bit of overlap, and there is still this peak, the contribution from the lipids. We may have a clear shot on the GLIC-A and the GLIC-B, and the one solution would have been to use a fancy deconvolution algorithm to try to estimate the contribution from those lipoproteins, which is pretty much always present, but with a different contribution. But if we look at the gray box on the bottom of the slides, you can see selective toxi that were performed show that the contribution from the lipoprotein is not easily modeled just by a Gaussian, but may have a different contribution that overlap with the GLIC-A. So also it would have been an improvement. It is not very easy to accurately model the contribution from those lipoprotein. Then the idea of having an experiment that leads to a clear spectra sounds much more attractive to us. So just to show a little bit the approach is nothing very, very fancy. The Dyer experiment that was proposed originally, and the modification that were brought to come with this JEDI. And as I've mentioned, it was mainly a trial and error, because at the beginning we were convinced that the driving, I mean, the filter came from relaxation, while it turns out it was the J addition that removed most of the contribution most effectively. The price to pay, so by going from Dyer to JEDI, we have a much cleaner spectrum, and we have twice the signal to noise. So this is really attractive. The only down the side to this JEDI is that we still have a pretty long T2 relaxation, since we have to wait for the proper defocalization of those multiplets. So we've tried to improve on that, but at some point we had to a good compromise between relaxation and defocusing. So now how can we evaluate the accuracy of this strategy? We look for proper cohorts, I mean real samples where we can demonstrate the improvement in accuracy, and we've tested with 2 cohorts. So again, the Spanish cohort, which ended up very handy and helpful, because we've measured a lot of experiment on this cohort, where we have COVID patients and control, and another very interesting cohort with 100 pregnant women and 7 time points. And this is a very interesting collaboration with University of New South Wales in Sydney, because those pregnant women actually were shown to have a huge increase during a pregnancy of lipoproteins. So mainly the peaks we are interested in minimizing. So as you can see here, the lipoproteins measured for these mumps, that's the name of the project, it's not my invention, the name of the project. We can see that in blue, the first trimester, and in black at birth, most of the lipoproteins parameters are strongly increased, and then eventually, as you can see on the one of the parameters I've just selected, after birth in red, we can see that most of the parameters are going back to normal after 6 weeks, 6 months, and then 12 months. And then again, just to highlight the strengths of the IVDR and the standard method, we could also select a subset of women from the COVID cohort that match the age and the BMI from this cohort in Australia, and just show how those women, we suppose, are not pregnant. Also 2 are a bit higher, maybe, Oscar. And then we can actually use control from different cohorts to cross check. So this is a work that is being done by one of member of the lab, Samantha Lodge. And if we look at the results, so the hypothesis is, if we look at the spectra, if we remove properly the contribution from the lipoproteins, then we should get a much higher correlation between GLIC-A and GLIC-B, as the lipoproteins alter the signal of, or the concentration, or say the integral, to make it simple, of GLIC-A more than it should affect GLIC-B. And if we look at, so these are on the left, all the samples measured for the mumps cohort, we see the ratio, I mean, just the correlation between GLIC-A and GLIC-B, with a pretty poor correlation actually. And then if we perform exactly the same, but now switching dia with this new GDI experiment, then we can get a much higher correlations, meaning that we convincingly remove the contribution from those lipoproteins. And if we look at the center panel, we can see the box plot obtained. So in that case, red means dia, black means the GDI information. And if we look on the first 4 box plot, we see again the healthy control versus the infected patients. We can see that by going from so the offset in intensity is mainly due because we are measuring just the integral. So there is no unit. And because we have an increase in signal to noise, then we have a higher integral. But we can see, most interestingly, is that the significance between the 2 groups is increased when we go from dia to GDI. But most important to us is the fact that if we look at the box plot for the infected patients, and we compare to the pregnant women at birth, we can see that with dia, the median are very similar, meaning that we would say that we would basically classify a pregnant woman as COVID positive by those standards. While when we switch to dia and get a proper estimation or more accurate estimation of GLIC-A, we can see that the box plot for the acute phase patients in COVID is much higher than the mum at birth. And we can see that the red box plot for the mums cohort is so the increase is much more pronounced on the dire box plot than it is on the GI, and this is mainly due to the contribution of the lipoproteins I've shown are very high and enhanced during the pregnancy. So by removing that contribution, we get a much flatter curve for the mums cohort when measured with GI. And then just on the last box plot, we can see what happens on the. We are interested now in understanding also all the outliers or the patients that were actually classified as outlier by one methods and not by the others, to check actually and to go back to the spectra and look to make sure that the contribution and what we are measuring is actually what we should be measuring. Sorry. So what comes next? I've just focused on NMR, but obviously we've compiled a huge amount of data, not only by NMR and for different cohorts. And obviously we are interested in making a few crucial links. And just to mention another very important parameter, which is the ratio AB-A1, so apolipoprotein B 100 over apoprotein A1, that is or was linked to the cardiovascular risk. And you can see then by compiling different cohort, in that case from Spain, from Cambridge and from Western Australia, we can see how first those data align very well together. So we can see in green the normal cohort, and we can see the two bars highlighting the level that are considered as healthy and critical. And we can see that even in the normal or healthy control, we still have people that by those standards are at risk. And what we were interested is to follow or to estimate if the infection by COVID would actually shift some of those population into a higher risk area. And indeed, we can see that the acute phase patients have much higher level of this ratio is much higher, in some cases very critical. And then on the gray box plot, we can see what happens after 3 months, and in some cases even after 6 months. And we can see that hopefully for us, in most cases, those levels are actually going back. But still, there is, even after 3 months and after 6 months, patients that are not going back, and those are paired data. I haven't the time to go into the detail, but we can show that fortunately most people go back to normal, but there's still a population which is at risk after a long period of time. Then obviously we are interested, as the GLIC-A was linked by Oddfor's group into the inflammation space. We would like to link and to understand a bit better what is the role of this SPC peak in the inflammation process. As I've mentioned earlier, we are interested to understand why this SPC peak actually decreases during inflammation, while the other markers like GLIC-A and GLIC-B all increases. The first steps that we are going to explore are also to try to understand and map against all the parameters we have by measured by, for example, mass spectrometry, lipids and other parameters, with the what we call SPIC-A and SPIC-B, which are mainly the contribution from the LDL and HDL particle, and see if we can make different use of this marker. So again, there is a lot of work to compile this information, and the next step is to show how this SPC peak is linked into a normal or healthy population, and see if we can link it to other inflammation process, not only COVID-19. then I would like to make a special thanks to the IPCN, so the International Phenome Center, which Oscar is a member, and helped us actually gather all those cohorts. I haven't mentioned all into detail, but at least the cohorts we've got from SIG BioGuinea, also from University of Cambridge, University of Harvard now, and also University from Australia. And for all the team, we are working in Australia, so a fairly small group yet for the number of equipment we have there, and that helped us put together those first and preliminary results about SPC. So thank you.
Thank you very much, Julien, for this very interesting and vibrant talk. I don't know. I have several questions. Like one of them, a very technical question, like this JEDI experiment, because time is a must here, because you have to measure many samples. Like as compared to the DIRE sequence in terms of sensitivity, how does it compare?
In terms of sensitivity, we have a factor of 2, as I've just shown. So we basically improve the signal to noise by a factor of 2.
So it means that it reduces the time by a factor of 2 as well, which
if we would like to trade it to time. Yeah, there is an ongoing discussion about what are the proper parameters, and this is something which is very important, because I may be blurred a little bit the frontier between targeted and just standard methods, because in that case we are not really making the link to the concentration. We are still comparing arbitrary units. So it means that in order to be compatible, or if we want to work with different groups around the world, we need to agree on a standard. Yeah, sure. So it's not really yet quantitative.
Mm-hmm. I understand. Marcus?
Thank you. I have 2 questions. The first one is, I mean, how much of a, let's say, optimization did you have to do between Oscar and yourself to make the data sort of being consistent in terms of running the experiments and whatever is required? That would be my first one. And the second one is, I think you didn't comment on it, but the outlook was to learn something about long COVID, I guess, and or what NMR could say to this. And could you comment on that?
Yeah. So the first, I would say there is no optimization between the 2 labs as soon as the 2 labs actually adopt these IVDR standards. So most of the optimization has been done by Bruker, and once the machines are actually properly set up and calibrated, with this IVDR, so mainly setting up the calibration for the erratic correction, we can compare them straight away. So we have not to do something additional. Now, the work that has been done with Oscar and with Bruker as a partner was the optimization of the new DIRE or PGP or JEDI sequence, because in that case, as Oscar mentioned, we have to negotiate and agree on what would be the optimum parameters, and then everybody should apply exactly the same. So that's the trade-off. We lose the possibility to use or to tune the parameters individually in each lab, but we gain the fact that we can actually compare data. And I think this is really powerful. And to be honest, that's the first time I really not only saw the results, but had the chance to join those data myself. And it's really amazing to see how we can join data from different cohorts, different labs, and without batch effects. It's truly surprising.
Yeah, that was even surprising for us. Like it works so well. So there is also a question from Genevieve Seabrook. Why not removing albumin from the beginning, and can you use 1.7 millimeter tubes for your study? Actually, two questions.
Okay, so I'm not sure what's the proper answer for albumin. So if we look at the NOESY spectra, and if we are interested into inflammation, albumin is actually the first markers that highlight. So if we take the control cohort and an acute phase, so COVID patients, and we model it using the NOESY experiment without performing any type of correction, so any baseline correction, anything, the first markers will be albumin. And if we look, and I'm not really a specialist, but I had to learn a little bit by talking to the clinicians that actually collected those samples. Albumin is a markers, and when they in the hospital test for or follow or treat the patients, they've tried to assess the inflammation process. So there is many different parameters like the CRP proteins, but albumin is also a clinical parameters that can be measured, and to some extent it is a parameters that we could access by NMR. I understand that some work has been done on that. I also understand that it's not trivial to measure the albumin content in a very robust manner, since it's highly concentrated, and it will be strongly affected by any problem with the water suppression. So there is probably technical issues, but I think it is also something very powerful, because the albumin may contain a lot of information when we are interested into inflammation process. So I'm not sure I would like to remove it like per se. That's for one. And then if it's just the process of removing the protein, I would say the CPMG is doing a great job at that. So I think if we don't have, and then for those kind of application, if we dream to translate into the clinical environment, time is really crucial. So if we can, I mean, in that Oscar mentioned the Bruker is interested in moving fast. It's because we would like to minimize the time between the sample is taken and the answer is provided. If we have to make more steps in the sample prep, then we are actually losing minutes to give results. And I think the strength of NMR is really when we compare that to mass spec, is it can be very fast.
Mm-hmm. And about the 1.7 millimeters?
1.7 millimeters. So yes, we've tried. Yes, we can measure. So we've done, but then the problem is there is no standard method for now. So we can obtain even with 70 microliters. So very, very low amount of samples. Very nice. Nice enough spectra to work with, but the problem is that we cannot extract and compare those with 3 millimeters and with 5 millimeters. So for a separate kind of experiment or project, it may work very nicely. But if we want to then use those data and merge them with another cohort, now we are unable to do that.
Mm-hmm. Okay. Another question from Lu Yun Lian says, are there independent biochemical validations that click A, click B are elevated? This is important since the NMR signal has not been specifically assigned in this spectrum. Well, I guess it asks for the validation on the click A, but this is clear, right?
Yeah, so that's the next step actually. So yes, indeed. So some work has already been done, and the linkage between the observed peak and the inflammation has been done by others, and that's precisely the last slide. So we would like to also be able to link not only click A and click B, but SPC, peak, this information. So that's why we are also interested in linking and making correlation with other clinical parameters that we could use as a validation. Yes.
But also you showed beautifully a correlation with the cytokines. I mean, it's very clear, the link with the inflammation, right?
Yes, no, definitely. But the thing is, this is just the next step. So that has to be validated on a larger cohort. And I think now we are reaching a fairly or reasonable numbers with thousands of points.
So I think that there are some people, and this is slowly merging into the informal session already, who would like to talk. I mean, I can see Armando Ariza Castolo. Perhaps you can formulate your question yourself. You're muted, Armando. Okay. Perhaps he doesn't want, I cannot see any other. So picking up Julien on Marcus' second question.
Yeah, I owe one answer.
I mean, you clearly showed, I mean, clinicians tell me that long COVID can be differentiated between pro-inflammatory, pro-atherosclerotic, and pro-neurological. And it seems to me that you clearly showed biomarkers for the first two. And then my question on top of Marcus one would be, do you think that we could also clinch the pro-neurological, or at least the neurological affectations on long COVID?
Yeah, so yeah, this is just the question a lot of people would like the answer, because actually we are all impacted and affected by these pandemics. So I've just shown one because the focus was mainly NMR, but of course we've measured by mass spec other what we call observation windows. So for example, metabolites related to the tryptophan pathways. And we can see, for example, that also markers like kynurenine, tryptophan, quinolinic acids will eventually come back, but some are very slow to get to a normal pace. So as I can show here with the ABA1 ratio, we can see that most people will recover or normalize after several months, but that we have a fraction of people that are not going into the correct direction. And we've compiled these very same figures for 600 parameters, and we can make a list and a panel. So that's what we are trying to work out to consolidate the first result we obtain, where we show that we have different risk from so neurological risk, cardiovascular risk, and the other what we associate actually with joint pain, etc. And those parameters are not going back to normal. Now, to be honest, I think one of the challenge now for us is to convert this analysis of outliers into something which is statistically meaningful, because as we can observe those parameters going up and eventually going down, it's not very easy to monitor those in a statistical approach that allows us to distinguish in what spaces those people are directed. So the more we stratify the cohort, the less statistical power we get. And we are, I mean, and then I welcome suggestion on that. We are a bit in an observation process whereby we have to make comments on abnormal behavior while all our statistical tools are mainly attuned to talk about the idiosyncratic data, but not about the non-idiosyncratic patients. So I think, at least from the data perspective, because I'm not a clinician, one of the big challenges is how to extract statistically significant data from what we are observing. So now I would have to show a lot of data, and we can observe actually by eyes that fraction is not going back. But now trying to link them into the different risk or the different medical condition is a complicated task. So what the next step we are taking in the group is to use a very big cohort of normal people pre-COVID, which was acquired and collected in Busselton. So 2,000 patients that were followed on different time points, so every 5 years, to try to see if we can actually make a proper classification on this normal cohort. What would be the markers for all those different spaces that I've shown in the first figure, and then use that to make prediction about the COVID patients. But in some cases, the links are already done by others. In the case of the ApoA1 ratio, it's linked to and accepted that it's associated to cardiovascular risk, but not all other parameters are as easily linked straightforward to a particular neurological or whatever dysfunction. So sorry for not offering a more clear cut answer.
Well, that's fine. I think this approach makes a lot of sense, and we're thinking it's something very similar.